Hello everyone. Let's get started. Today we're going to be discussing the acellular microbes, the viruses. By definition, a virus is a parasite. It's an obligate intracellular parasite, and it's composed of nucleic acid surrounded by protein. Viruses cannot replicate themselves, and they cannot perform any metabolic reactions. Viruses are quite simple in their structure. They contain a piece of genetic material, either DNA or RNA, that is surrounded by a coat of protein. We call that protein coat a capsid. And when we think about the genetic material and the protein coat together, we refer to it as the nucleocapsid. Viruses are not cells. Remember how we defined a cell. Viruses have no cell membrane, no cytoplasm, no ribosomes, no organelles, none of the things that we associate with cellular life. Because they are not cells, we refer to viruses as particles, and we call the particle, when it exists outside of a host cell, a virion. Because viruses are not cells, they are technically not living things. They have no ability to replicate themselves. They have no ability to metabolize. They cannot take in nutrients and expel wastes. They have no ability to receive information from their environment or integrate and respond to that information. We don't consider viral particles to be alive. They are obligate intracellular parasites. Obligate meaning they must get into a host cell in order to replicate themselves. They're parasites because they rely on the mechanisms of the host cell to do the things that the viral particle needs in order to persist in the world. The host cell is going to do the work. The host cell is going to make viral protein and more viral genetic material and do the work of assembling new viral particles. It is a true parasite. Now, viruses have to get inside of the host cell in order for all of this to happen. They have to gain entry by interacting with receptors on the host cell membrane. That first interaction is crucial. They have to bind to the host cell membrane in order to get their genetic material into the host cell. That genetic material then has to be replicated. More copies of it need to be made. And it also needs to be transcribed and translated so that viral proteins can be made. Once this work is done, new viral particles can be assembled in the host cell. New nucleocapsids, in other words, can be assembled in the host cell. I want to spend a few minutes talking about some of the details of viral structure. One of the ways that we classify viruses is by describing them as being either naked or enveloped. Now, a naked virus consists of the things we've discussed so far. It's simply a piece of genetic material, DNA or RNA, and the protein capsid. Many of the viruses that infect plants and most bacterial viruses, yes, bacteria can be infected with viruses, are naked viruses. Enveloped viruses are a little bit different. Enveloped viruses consist of a piece of genetic material and a capsid coat, but they also have a layer of membrane what's referred to as the envelope, around the nucleocapsid. The envelope is actually acquired from the host cell. As an enveloped virus exits a host cell, after new copies have been produced, the virus exits the host cell through a process that looks a lot like budding, the way yeast cells bud. As the viral particles bud out of the host cell, they take a piece of the host cell membrane with them. Enveloped viruses also tend to have spikes of protein emerging from the envelope. These spike proteins are viral proteins, not host cell proteins. Many of our animal viruses are enveloped viruses, including the virus that is causing the current pandemic. In this slide, we can see some drawings of naked viruses and enveloped viruses. On the left-hand side at the top, you can see a typical naked virus. They've shown us the genetic material in purple as a twisted strand, and then they've drawn a coating of yellow circular subunits that are proteins composing a capsid. You'll notice that there are sort of these purple spikes coming off of this naked virus. Those are viral proteins. On the left at the bottom, there's another virus that would also be considered a naked virus. It just has a different shape. You'll notice that the genetic material, again shown in purple, is wound into a helical coil this time, instead of that jumbled structure we saw above. There's still a coating of these yellow proteins, so we would refer to this again as the nucleocapsid. On the right-hand side of the slide, at the top, we can see an enveloped virus. The genetic material again is shown in purple, jumbled up in the center of the particle. They've cut away most of the structure of this viral particle so we can see into the structure. The yellow circles of protein, again, are surrounding the genetic material. But now we have the addition of this purple circular structure. This is the envelope. This is actual membrane, and it was acquired from the host cell. And then these blue structures that are sticking off of the envelope, those are spikes, spike proteins that are viral in nature. Down at the bottom on the right, we can see a viral particle that looks very similar to the one on the left. The only difference is it now has an envelope around it. So, viral particles have genetic material inside of them. Sometimes it's DNA and sometimes it's RNA. That genetic material is surrounded by a coat of protein that we call a capsid. The protein coat or capsid is often composed of a limited number of proteins, sometimes only one protein that repeats over and over and over again, creating that capsule. The individual repeating units of that same protein are referred to as capsomeres. It's in the best interest of the viral particle to have as simple a structure as possible, because the more simple the structure is, the easier it is for the host cell to replicate it and rebuild it inside its own cytoplasm. A good example of the simplicity of the protein coat comes from a virus called the tobacco mosaic virus. This is a virus that infects the tobacco plant, as well as other plants like the tomato and similar varieties. The tobacco mosaic virus contains a single strand of RNA as its genetic material and one protein that has been copied 2,130 times. Yes, we know exactly how many capsimeres create the capsid around the tobacco mosaic virus. One capsimere protein, 2,130 copies of it. Over here on the right-hand side, you can see a drawing of the tobacco mosaic virus. So this is one of those elongated viruses that we saw on the last slide. You can see that that strand of RNA is coiled up and surrounded by, again, over 2,000 copies of this one capsimere protein, shown in blue. Viruses have different shapes, the same way that bacteria have different shapes. Viral shape is usually very symmetrical, and it looks quite different from what we see in bacterial cells, in living cells. There are two what we call basic shapes of viruses. There is a rod shape, like the tobacco mosaic virus, and there's what we would call a spherical shape. Now, the spherical shaped virus actually comes in two varieties, depending on whether or not it's enveloped or naked. There is a truly spherical virus. Some of our enveloped viruses, like the virus that causes COVID-19, are spherical viruses because the envelope of membrane makes them true spheres in 3 dimensions. But some naked viruses, while they look spherical from a distance, are actually polyhedrons. They're actually composed of triangular faces that come together to create a generally spherical structure. And we'll take a look at these in just a minute. Besides the two basic shapes of viruses, there are also what we call complex shaped viruses. These include the bacteriophages and the giant viruses. A bacteriophage is a virus that can infect bacteria. And when you look at a bacteriophage under the microscope, as you can see over here on the right-hand side of the slide, they don't even look real. They look like some artist's imagining of an alien creature. They've got this sort of head structure, and then a neck, and then these spindly sort of legs and feet. They honestly don't look real, but this is exactly what they look like under the electron microscope, as you can see in this image. So this is considered a complex shape, much more complex than that simple rod or sphere. The other complex shape we see are the giant viruses. Giant viruses are actually the newest type of virus that we have discovered in nature. Giant viruses have likely been around for a long, long time, but they were only discovered in recent decades. They were originally believed to be some form of prokaryotic cell because they look a little bit like a prokaryotic cell under the microscope. They're large, hence the name giant, and their structure is something similar to cellular. However, it's recently been discovered that these are not cellular life forms, but viral particles. And they've been discovered to infect a particular type of single-celled eukaryote, the amoeba. There are a handful of giant viruses that have been investigated so far, and they have names like the Mimi virus, the Mama virus, and the megavirus. These terms Mimi and Mama and Mega are actually abbreviations for things. That's where the names come from. On this slide, we can see some more artists' drawings of the different viral shapes. In the upper left, you can see, again, a spherical shaped virus, but it's got the true polyhedron form rather than a true sphere. You can see that this structure is actually made up of triangular plates that are assembled together to make this, again, somewhat spherical shape. You can see the spike proteins emerging off of the nucleocapsid as well. This happens to be a drawing of what's called an adenovirus. This drawing represents an influenza virus, and this is a true spherical virus because it has an envelope around it. Down here, this is a rod-shaped, or what we also call a helical-shaped virus. This is our friend, the tobacco mosaic virus. And over here on the right, this is a bacteriophage with its complex shape. Bacteriophages are truly fascinating. The genetic material is enclosed in this protein capsid up at the top. Then there's this sort of a neck structure that functions just like a hypodermic needle, a hypodermic syringe functions. These little spindly legs at the bottom do the job of contacting the host cell membrane and interacting with receptors. And then I think you can see there are these little sort of spikes here. Those will contact the cell membrane and anchor the bacteriophage in place. And this central portion will then compress just like a syringe and inject the genetic material out of this head and into the cytoplasm of the host cell. On this slide, we can see some more drawings of viruses and actual electron micrographs of what these viruses look like. So here is, again, a rod-shaped virus or a helical virus. And this down below is an image taken with an electron microscope. This is our friend, the tobacco mosaic virus, a helical or a rod shape. Over here, this is one of the spherical shapes. This is the polyhedron. This is the adenovirus once again, and this is how it looks under the electron microscope. This next drawing represents an influenza virus, a truly spherical virus, and this is the influenza virus under the EM. And here again is a bacteriophage, a virus that infects a bacterial cell, and this is what it looks like under the electron microscope. Viruses are exceedingly small, and it's important for us to remember that they are much smaller than bacterial cells. Generally, the range of sizes for viruses is between 30 and 300 nanometers. So remember, viruses cannot be seen under a light microscope. Bacterial cells that are seen under a light microscope look very, very small. Viral particles simply cannot be seen using that type of microscope. If you look over here on the bottom right, you can see a nice drawing that reminds us of just how small viruses are. The largest structure here is representing a eukaryotic cell. This represents a bacterial cell. And then we can see viruses. And you can see that viruses do range in size. Again, 30 to 300 nanometers. Very, very small particles of material viruses are. Giant viruses got their name because they don't fit the normal size range that we expect for viruses. Giant viruses are actually more in line with the size of a bacterial cell at 400 to 1500 nanometers. So remember, a virus that is 1500 nanometers long is 1.5 micrometers long, and our bacteria are typically 1 to 2 micrometers in length. So giant viruses truly are giant. This drawing I put in because it reminds us again of just how small viral particles are in comparison to living cells. What you're looking at in the drawing is a representation of just a little piece of a gram-negative bacterial cell. So we just took a little sliver out of the membrane of this gram-negative bacterium. Here's the plasma membrane with blue heads and gold tails. You can see some purple proteins inside this plasma membrane. Then we have the periplasmic space in white, and then a thin layer of peptidoglycan shown in gray. That's the cell wall. Then we have the outer membrane that is only found in gram-negative bacteria. And remember the lipopolysaccharide, the LPS that comes off of that outer membrane. They've shown us that this particular cell has a flagellum. They've cut it off here so we can't see it in its entirety, but see how big that flagellum is. It also happens to have a pilus here. And over here we see a bacteriophage. So if we were to imagine the entirety of this bacterial cell, it would be enormous. This is just a little sliver of its membrane. And here is a bacteriophage in comparison. Viral particles are very, very small.